As Monica said, I'm Francie Barron. I'm the Vice President of Innovation Partnerships here at Cardea Bio. Just a little bit about me. I am a biologist. So I got wrapped into working for a semiconductor company about 9 years ago because I know the CTO, Brett Goldsmith, from college. And he had this crazy idea of taking these biosignal processing units and creating the next generation of diagnostics. And after my postdoc at Stanford, I was doing regulatory affairs, and I said, well, you need to go to the FDA. So that's kind of how it all started. So I joined the team first as a VP of R&D and really helped with the beginning understanding of our BPUs. And then I switched over to business development about 3 years ago. So my job and my role is really to interface with people who are interested in using our biosignal processing units, help them to understand the technology and understand how we can create products together that can revolutionize different markets. So today, I'm going to talk a little bit about the technology itself, how we kind of got here, why we got here, and our vision. I'm also going to kind of focus in on one of our applications, which is amplification-free genotyping. And then I'm going to end with kind of a broader vision about how BPUs can be utilized in a variety of different areas. So I like to start out with kind of how I learned biology, right? In biology, we had the omics streams. We reduced everything in the cell to its individual parts. We have genomics, transcriptomics, proteomics. You put your favorite words and add an omic at the end. And we learned a lot. We were able to understand the different parts of the cell and what they kind of did, but we weren't getting the whole picture. We were taking snapshots. We had to really break open the cell and not really understand it in its context. And we know, right, that biology is a dynamic science. It's a dynamic living thing that constantly changes in response to the environment. And so when we think about this mass of data, we want to try to understand it not in its individual parts, not in the single omics and snapshots, but in a more dynamic or systems biology approach. But that's a lot of data, and especially a lot of data to process in real time. So one of the things that we understand is that there's a gateway that we need to make between biology, a bridge. That bridge, we think, comes from nanotechnology. So I can tell you over the last 7 years or so, I have learned a lot of physics and engineering in my lifetime. And I'm seeing how this bridge, this gateway, this connection between these very dynamic and complex omic systems are being connected to the computer and electronic worlds that we live with today and rely on. These computers that process all of this information, we can distill down into a single chip, and understanding that this is what we use to process the information, to gather it. But there has been a disconnect with our ability to leverage our computers and connect that to biology. So before I get into how we created this gateway, I want to talk a little bit about the history of transistors. So in the early 1900s, we first started out with vacuum tubes, and that really transformed us. We were able to now communicate with each other via phones. And it wasn't until the mid-1900s that silicon transistors were first formed. And what was great about this is it enabled us now to move and process information much faster. That allowed us to make the first personal computers and personal phones and devices. And about 20 years later, those transistors were made into integrated circuits. And that was another jump forward in our ability to do computer processing. That's what enables everything that we rely on today. The downside to silicon, so while it's allowed us to do all of these wonderful electronics today, it hasn't allowed us to connect to biology. And that simply comes down to silicon does not play well with biological fluids. And you probably know that if you've ever dropped your phone into the ocean. So we needed a different semiconductor material other than silicon to enable us to create the first biosignal processing unit, which we did around 2019. So the semiconductor material in question is graphene. Graphene, if you haven't heard of it, is a single atomic layer of carbon. This is a biocompatible material that enables you now to function in salty solutions, which means that we can start to interrogate the different multi-omics biosignals that are in life around us. The other great thing about graphene is that because it's a single atomic layer, it's a super fast, near perfect semiconductor. So it allows us to have the time resolution that's required to watch these biointeractions in real time. So beyond the chip, we really have a platform. And so I wanted to take a moment to break that down into how we think about it. The BPU platform consists of 4 major parts. The first is the wetware, which we're going to talk a lot about today. That is how we're going to recognize our analytes. That forms a biolayer on our nanoware, or the BPU itself. We connect those BPUs via our hardware so that we can read out those electrical signals, translate them, and put them into our computer. And then finally, we have all the software that ties it all together. It takes those electrical signals and processes them in a way that we can interrogate the information and make conclusions about the system we're looking at. So I want to zoom in to the wetware and the nanoware, because that's where all the magic happens. When you zoom into our consumable chip, which you can see that we mass produce today, thousands of chips now per week, you can see that we have a BPU pattern that you can see here. So what you're looking at is 3 channels. That means that every single plex chip that we have has a replicate of 3 measurements per chip. And the little rectangles that you see here represents 5 transistors. Those transistors are made of graphene and where the biomolecule is attached. They're run in parallel, and we have them mostly for production redundancy. So if any one of those breaks, we still can make a measurement happen. If we look down further at a single transistor, which is depicted here, you can see we have our graphene layer, we have our antibody and our analyte in solution. There's 2 currents that we apply, and I'll explain a little bit about what that means later. First is a liquid current that is applied within the solution that you've placed onto the chip, as well as a current that goes through the graphene itself. So the transistor principle, it can be broken down fairly simply into this idea that when you have a biolayer on the nanoware or the graphene layer, it becomes a part of that electric circuit. And as you flow electrons through it, you're going to get an electric output. We take that measurement, and then when we place our biosample on it, whether that might be serum or saliva or just a buffer, our analyte is going to interact with the capture mechanism. And if it binds, it's going to cause a disruption in that signal or an electrical change that the graphene can very sensitively pick up. So I've talked a lot about how we believe that our platform is a true multi-omics platform, but what does that actually mean? We are able to conjugate a wide variety of capture mechanisms onto the graphene. We can do receptors, we can do proteins, antibodies. I'm going to focus a lot on CRISPR today, but you can also do aptamers or peptides. Basically, if you can bioconjugate something to a surface, you can conjugate them to our BPUs. And that is what drives the specificity and the sensitivity of the system. We can't make magic, but if you have a good antibody, you have a good protein, you can pick up a wide variety of analytes. So while I am going to focus on CRISPR today, I do want to kind of give you a taste of some of the other data that we have published on and taken in our labs. So the first is immune biomarkers. IL-6 is a cytokine. In this experiment, we have compared our BPU in a traditional ELISA format compared to an off-the-shelf IL-6 ELISA kit. We're using as our sample a conditioned media where IL-6 has been spiked in. There's two things of note. One is that our lower limit of detection is pretty comparable to the ELISA. And the other is that our dynamic range is actually much larger. So you can see here that it doesn't actually level off. The other big comparison is that the ELISA took a couple days to run, and we got all of this data in a single day. Another example is a publication in Advanced Biology, Advanced Biology, back in 2021. And this is where we're looking at an exosome comparison on our chips, also compared to an exo-ELISA. Now, one of the major differences is that the exo-ELISA actually requires a lot of exosomes in order to do a very nice standard curve, at least up to 10 to the 10. We can actually use a much smaller volume and still get sensitivity down to 10 to the 4th. Early days when we were working with Zika and with the CDC, we also did a publication looking at utilizing one of CDC's developed antibodies and comparing the detection of an antigen, non-structural protein 1 for Zika, as well as a negative control Japanese encephalitis. What you can see here is that in just as little as 5 minutes, we're able to distinguish between the positive control, our Zika virus here, and a common interferent, Japanese encephalitis. More recently, with the SARS-CoV-2 outbreak, we had a collaboration with GTRI, DARPA, the Army, and Siemens Healthineers, looking at collection of aerosols and detecting of SARS-CoV-2 virons. We also looked at detecting them in saliva. So the graph that you can see on the right shows that we can specifically detect SARS-CoV-2 while not detecting H1N1 or rhinovirus. So I'm going to move now on to the CRISPR chip. Now, the CRISPR chip, if you're not familiar with CRISPR, but considering you guys are mostly in the genomics field, you probably have heard about it. It's a wonderful little enzyme that can open up DNA and search for a specific sequence. So at Cardea, we very much believe that technology or biology is technology. And so we utilize this like a search engine. So if you can reprogram or create a guide RNA to a sequence you're interested in, then you can look for it utilizing CRISPR in an amplification-free methodology. We actually published this in 2019 in Nature Biomedical Engineering, where it was one of the most read papers of this year. So I love this movie because it dives a little bit deep into our chip. So we're zooming in now to the 3 channels that I talked about earlier, highlighting the 5 transistors. And deep down in, we're bioconjugating the CRISPR molecule to the graphene surface. As I said, CRISPR unwinds the DNA. It's programmed by that guide RNA, and it can search and find the gene that you're trying to detect. So the publication in 2019 focused on Duchenne muscular dystrophy, or DMD. Specifically, we looked at 2 exons, exon 3 and exon 51, both which cause DMD. However, some patients have exon 3 and are deleted 51 or vice versa. So in the graph that you're seeing here, we have a healthy where the exon is present. And we have Duchenne patients where that exon is also present. However, in these 3 on the right, it is not present. You can easily see the inability to detect versus the ability to detect. We also looked at a linear curve of our ability to sense, looking of exon 51. And our lower limit of detection in this assay is 1.7 femtomolar of genomic material, or approximately 1,000 copies per microliter. So we did all of this with a simple buccal swab from DMD patients. The following year, we also wanted to take this a step further. So instead of looking at a gene and the lack of that gene, could we detect a single SNP? We chose two conditions in order to look at this: sickle cell anemia and ALS. In this case, we were able to detect very easily the presence or absence of the sickle cell, and the same with the ALS SNP mutation. We did a lot of this work with Dr. Virginijus Siksnis at Vilnius University and Dr. Phil Collins at the University of California at Irvine. Also in this publication, we were able to look at heterozygosity in sickle cell patients. So we could tell whether or not they had a single wild type gene and a single mutant gene. Now, I've focused a lot on genotyping. You've probably also have heard of many different other types of CRISPR-based diagnostic methodologies. And I love this review that was done last year where, and of course, I'm biased because our CRISPR chip is mentioned. And while the thing that I'd like you to take away from this is the fact that we are the only technology that has been published that does not have amplification. This really enables you to start taking your applications outside of the clinical lab and bring them closer to the point of care, point of testing. And it's not just in the clinical space. We actually also have a partner that is in agriculture. So not only in the doctor's office, but also in the field is a desire to look at transgenes. With food traceability becoming a much more dominant need in the agricultural industry, there's been a huge push to move towards being able to easily and quickly identify transgenes in the field. So what you're looking at here are two different transgenes from a plant. And you can see very easily that we can distinguish between the wild type scenario and the transgene in both of those. So today, I'm not going to talk a lot about our data that is behind CRISPR-QC, but I am going to mention that CRISPR chip is being utilized by CRISPR-QC for gene editing and quality control. Now, quality control methodologies in the CRISPR space are very few and far between. And in fact, there are yet no standards by which we can tell if our gene editing experiments are doing well. And since they are now in clinical trials, there is a huge need to create these standards. So CRISPR-QC, in partnership with Synthico, NIST, or the National Institute of Standards and Technology, and the NIH, are working together to actually establish these quality control metrics. So Ken today will tell you a little bit more about CRISPR-QC and its commercial use of the CRISPR chip, and also the data behind things that he can do. Now, I started out talking about single omics and how our chip can be a single platform for multi-omics. And so I challenge folks all the time to think about what impact a true multi-omics platform can have. Cardea has thought long and hard about this. And one of the areas that we think we can bring a lot of value is actually in the space of cancer. So as you might know, cancer has a lot of biosignals. They can be found in the tumor microenvironment, but they're also shed a lot into our bloodstream. And while most biopsies can yield a lot of information about our tumors, they tend to be invasive and they have a very small amount of quantity of material. Blood is a lot easier for patients to give, and it still has a lot of information. Additionally, a lot of the current technologies that are out there that are interrogating these liquid biopsy signals are not able to look at all of them. They're much more single omics focused. Not to mention, if we were going to do it, the cost of running all of these largely sequencing based or ELISA-like platforms would make it very difficult for any patient or doctor to have that test on a regular basis. So we believe that Cardea's platform has established its ability to look at things like immune responses, exosomes, and DNA utilizing our CRISPR chip. And so we can use that on a single platform to enable a very quick way of looking at cancer biosignals within the system. Additionally, because we're based on electronics and not instrumentation that requires optics, it means that we not only get the economies of scale as we increase how many chips that we make, but we also can make very small readers that can fit into anybody's home or doctor's office. So I'd like to say that our Cardea BPU really can enable an all-in-one platform for next-gen liquid biopsy. And this is a flagship application that we're very much excited to be going into. So I wanted to, of course, acknowledge everyone and their hard work into creating the data that you saw today, as well as our products. So on the left is our Cardea team, both past and present. And on the right is our Keck Graduate Institute, which is Kiana Aran's, our CSO's academic lab. We also have a wide variety of partners that we are working with, and I wanted to take a moment to acknowledge them. So with that, I thank you, and I'd be happy to answer any questions.